Thank you so much, Valeria. Thank you for the introduction and for organizing this, and especially for putting together a very interesting flyer. And thanks everybody for coming. So as the title says, I'm going to talk to you about a topic on copy number profiling. Now, I do want to mention that the papers on this topic from my lab have mostly been published. So they do not necessarily represent the most current research that is ongoing in my lab. But the reason I choose this is because I think the biology here is much more straightforward. It's easier for me to explain in a short period of time, and it's easier for you guys to follow. And I think the whole purpose is just to give you guys a sense of the type of research that we do, and I think that serves the goal for today's talk. Okay, and feel free to stop me whenever you have any questions. So copy number variations or insertion or deletions and duplications that lead to either gains or losses of large segments of the chromosome. They are an abundant source of variation. If you look across different subpopulations or different ethnicities, you see that we have these polymorphisms with regard to the different patterns of deletions or duplications across the different cohorts, okay? And then these are what we call the germline copy number variations. And then these are inherited from the parents, and then they really don't result in a very complicated consequence or phenotype. So basically, these are naturally inherited across the population from the parents from generation to generation. And on the contrary, shown here is a very extreme case where I show the copy number variation in a tumor sample. Now, this is a case where we call the CNVs as the somatic copy number changes. So in other sense that you don't get this from the parents, but rather these are developed in the cancer samples, okay? And then you see that you have very, abrupt changes. Sometimes you lose entire copies of the chromosomes, and sometimes you get maybe 2 additional copies for whatever reasons. So the cancer genomes are actually very messed up. And what's really interesting in this case is that these patterns are only exhibited in the cancer samples. You don't see this, for example, in the normal blood samples. And then, as I'm going to elucidate more later on, different parts of the cancers or different clones of the cancer or different subpopulations of the cancers actually have different patterns of the copy number variations as well. And this is the main reason why cancer is just so hard to treat. Sometimes you have the bottleneck, but it doesn't necessarily kill all the cells. Only certain cells are very effective with regard to the cancer treatment. Okay, but more on this later. Here is just an overview slide of CNV in the natural setting as well as in a disease setting. So it is very important for us to be able to detect the CNVs on the genome-wide scale, with both high sensitivity, you want to call most of the variants that are there, but more importantly, you also want to be very specific. So you want to have high specificity. You don't want to have inflated type-1 error in some sense, especially in the clinical setting, okay? So how do we do that? Here are the techniques that I'm going to focus on today. We're going to start with bulk DNA sequencing. So these has been done for decades. You take a lump sum of the tissue, and then you just homogenize them, and then all the DNAs are mixed up together, and you send it to sequencing. And then I'm going to show you the protocols of the sequencing so that it's hopefully not a jargon for most of you, okay? And then just during the past several years, the field has really evolved into single cell sequencing. So I'm going to talk about our work on extending the framework from the bulk tissue DNA sequencing to single cell DNA sequencing, okay? So we'll start with bulk DNA sequencing. And then we're going to focus on whole exome sequencing, which still is very effective and very popular nowadays. So what are exons or the exome? So exons are the quote unquote functional protein coding regions of the genome, and they account for around 1%. So you have a piece of the DNA, and then a large chunk of the DNA are non-coding. Some people call it a junk region. And then only the gene regions are coding for proteins. And then even further within the gene coding regions, the DNA is, split into, consecutive introns and exons, okay? And only the exons are actually kept after the translation to be translated into proteins, okay? So eventually, basically the exons are most directly consequential or the most functional units of the DNA and encode for the proteins, okay? So as you can see, when we sequence the 1% of the genomes that are really key essential compared to the whole genome sequencing where you presumably uniformly sequence the 3 billion base pair of the genome, whole exome sequencing is very effective. It is cheaper. And it's still very popular, as I shown here. This is what I took just 2 days ago from the UK Biobank data. Some of you might have been exposed to it. Now, just back in last year, they have sequenced almost 500,000 of individuals. They do the whole exome sequencing of all these individuals, and the goal, at least for this part of the data, is just to develop a method that we're able to detect the copy number variations using this large amount of whole exome sequencing data. Okay. Now, before any of the modeling actually makes sense, I want to very briefly go through the protocols of whole exome sequencing. Now, as I said, the DNA is composed of coding, non-coding, intron, and extron. Okay, so the first part of the protocol is just to fragment the DNA into smaller pieces that are easier to be captured, easier to be handled. Okay, and the second part of the exome sequencing, they have these pre-designed arrays, hybridizing arrays, so they can uniquely capture the exonic regions of the DNA. Okay, so that you can anneal the DNA after the fragmentation into this, and you can wash away whatever that is not annealed to it, so that you can capture those that are kept, and then these are your exonic regions. Okay, and then what you do next is that because the DNA only have 2 pairs, one from the maternal side, the other one from the paternal side, you have to do some amplification for the sequencer to be able to pick it up. And then you finally do the sequencing in this case, okay? And then the sequencing, if you have not worked on this before, naively, you would think, okay, I'll just read out the A, T, G, and C, a sequence of that. But that is not how it is done. There are a lot of technical reasons why that cannot be, done in a straightforward manner. But rather, what we do is that we sequence very short pieces of the DNA. Okay, and then these are what we call the reads, and the reads are shown here in different colors. And then the reads are typically around 100 base pair, 200 base pair long. And then what you do is that you map the reads back to the genome, okay? And then we map it in this illustrative cartoon because you only capture the exons. All these peak regions where the reads pile up are your exonic regions, okay? And as you can imagine, if I have a deletion, let's say, then on average, I would have less reads that are mapped back to the exonic regions. And then, on the contrary, if I have a duplication, I will on average have more reads that are mapped to the regions that is captured. Okay, but that is all under the null. And then, but the biology is never that simple because along all these different steps, there are various bias and artifacts that are introduced. For example, some part of the DNA, it might be easier to be fragmented compared to the other ones. Some of the exons, depending on their sequence complexities, they might be easier to be captured. You have some sequencing artifacts and biases, so on and so forth. So what you truly observe is not, a real reflection of the copy number states. Okay, so but rather it's a mixture of the noise as well as the copy number true signals in terms of the number of reads. Okay, so I'll talk more about that. So basically, what we want to do is just to use the number of reads to detect the copy numbers. And then what we, the underlying, intuitively, how do you understand this is we just want to use the number of the times of a region that is read, as an analogy of how many copy numbers there are, okay? But the problem, like I said before, there are a lot of biases. The first one is a GC content. And then a little detour here, that the DNA is composed of A, T, G, and C. Okay, this is the last one, so bear with me. And then, different regions of the DNA actually have different content of the G and C. And then depending on what the content of the G and C, there is a differential ease of fragmentation because we know that the ANT is bounded by 2 hydrogen bonds and GNC are bounded by 3 hydrogen bonds. So it's easier to be fragmented in certain regions versus the other ones. Okay, so GC content is one source of variation or bias that we are very aware of. There is this capture and amplification efficiency differences, as I mentioned before. There is some batch effect. If you look across different population, there is also population stratification. But of course, there are some, other latent factors. These are basically the ones that you cannot directly quantify or you cannot directly model or measure. Okay, so how do we do? The input data, like I said here, is a matrix. Okay, the columns here are the samples. The rows here are the axons. So each of the axons you try to capture through the protocols that I mentioned. And then the color here corresponds to the number of reads. So the brighter here, the more reads, the more short reads that are mapped to that axon in that sample here, okay? But obviously you see a lot of low rank, factors that are presented even by, visualizing these two-dimensional heat maps. And obviously they have nothing to do with copy number. Hopefully I've already sort of give you enough motivation why they don't necessarily represent the copy numbers just because of all the bias and artifacts, okay? So what people do is just to apply PCA, individual. You can do that, a cancer you can sequence multiple times, but maybe that's not, you can think of these are independent individuals for now. No, yeah. But still you see this, low rank noises, and then especially the row factors that are shared across all the individuals. Yeah.
I still didn't understand how this copy number was. So in the very first picture that you showed, so in the chromosome, like with the normal and the abnormal. So what's the relationship with the copy number?
I'm not sure I got your question, but you can have some deletions or duplications along these axons, yes. Okay? In some individuals, you don't know which those are, and that is a goal. They want to recover which of these axons present a deletion or duplication in which subset of samples, but you don't truly observe those. What do you really observe? You can think of that as a surrogate of the copy number. That is the number of reads that are mapped to this. Under an ideal situation, the number of reads should be proportional to the copy numbers, because the more copies you have for a DNA, the more reads you should expect to be mapped to that. But you don't see this because it's not a good surrogate, because you have a lot of bias in the artifacts. For example, you see this stripe that is very bright here. It doesn't mean that all the individuals has a duplication for this axon. It's just only because the axon is longer. And the longer it is, the more reads that you get it mapped to the axon. So this is a low rank artifact that is presented in the data. And again, it has nothing to do with copy number. And our goal here is just to base off of this matrix, we want to recover what is a true underlying copy numbers. Okay, is that clearer? Okay, so people just use PCA. We extract the first few PCs, and then we expect, okay, the CNVs, lie in the high rank space, and then we recover, the low rank one, and then we complete it, and hopefully the CNV will pop up. Okay, question? Okay.
I see there's like this little up at the top there. There's some bright red.
Yeah.
So is it like variation within one of the horizontal lines?
Yeah. Well, I think that's because this axon is always captured much higher compared to the other axon, but also we have a column factor. These samples also are captured much deeper compared to the other ones. And then the column factor in this case, the white stripes here, again, it doesn't mean that this individual has the whole genome amplified. It can't be true, okay? It's only because we spend more on this individual and sequence more reads. Okay, so more reads are mapped to that individual, okay, across the entire axons. Okay, but the PCA isn't effective because it assumes a continuous measure, but here we have a read count integer valued input, assumes Gaussian noise structure. And then, so I'm going to show you later, the GC content is completely just ignored, but we can actually specifically model that. And we do have some biological priors in terms of how we should the most effectively model the GC content bias. And more importantly, the GC content, as I show you later, cannot be captured by a linear PCC in this case, okay? So this is what we come up with, what we call the Poisson latent factor model. We denote Y as the recount for exon I, individual J, okay? And then we model it with a Poisson lambda IJ, okay? And then the lambda IJ is under the null case. Forget about the CNV. So under the null, there's no CNV, what's really going on. All that we care about is just to decompose the lambda so that hopefully we can account for the over-dispersion of the Poisson. So how do we do that? The lambda is decomposed into different components. The first component, NJ, for the sample J, is what I mentioned is how much you want to sequence each individual. It basically tells us the total number of reads per individual, okay? How much money you want to devote into that. Beta I is what I mentioned, the length of the axon, okay? So the longer the axon is, the more reads will be mapped into that. The fj of GCI, this is a function actually for a specific individual. And then we know that it is a function that is of a shape that is shown here, okay? And then various different studies have shown that it certainly is not a linear function. It is probably a parabolic function, but most likely you should use a non-parametric version to capture the GC content bias in this case, okay? But regardless, it is certainly not linear. And then, you can directly sort of try to specifically model this so that you can attribute at least that part of the artifacts to the GC content. So we apply this, but we still see some residual artifacts that obviously have nothing to do with copy numbers. So it turns out you do have to incorporate these quote unquote latent factors. These are, again, the ones you cannot directly model or capture. And then we have K latent factors correspond to the axons, and then K latent factors correspond to the samples in this case. Okay, so these are incorporated in the data. And again, everything shown here is for the whole purpose of trying to capture the artifacts. And then once you capture the artifacts, you can estimate the lambda hat. And then this is the lambda hat that you expect under the null, the mean for this Poisson distribution under the null. But what you're truly observing in this case is actually deviation from the null, and then that is a copy number in that case, okay? So this is the model that I talked about. Again, this is a little bit of old, but this is the first piece of the work that I want to show some extension upon. And then just last year, this paper from Rafa Irizarry's group from Harvard Boston and Dana-Farber Cancer Center, they apply a very similar, if not exactly the same model to spatial transcriptomics data. And then in this case, you see it's still a Poisson data, and then you still have these latent factors that are shown there. They have alpha. I think I and J are kind of switched, between these two different ways of parameterization, but it certainly shows some resemblance between these different methods. Okay, so these are the GC content, the empirical fit from the GC content. I just want to show you what they look like. And I think in practice, we just fit a smooth line, I think, to capture the GC content bias. And then these are the ways that we adopt to choose the number of latent factors. These are very important, especially in practice, because there's always this trade-off. Okay, the larger your K is, the more cleaner you QC your data, but also you remove a lot of the true signals in that case. Okay, so it turns out that in this case, the BIC seems to give us and the number of latent variables equals to around 4, but you can also resort to the other ways of choosing the dimensionality of the model. Okay, so everything is compiled. These are the raw coverage. You do the normalization. You remove all those biases and artifacts that I mentioned. And then now you see this region that obviously have a nested deletion structure. And then we know that these are our positive controls, okay? So you zoom into this region, you tune your algorithms, and then this region is a positive control, and that is very important for genomic research. And the next step is segmentation procedure. You basically want to, segment the DNA into a region of the, into regions with the same copy numbers. Now, in this case, we use circular binary segmentation, but you can also use a hidden Markov model, for example. Yeah.
I think the processing goes into generating these graphs as they have the columns ordered in a way we can actually identify this state region. Are people grouped by some calculation structure, or are you basically taking the commuted graph and then saying, okay, find the permutation that
that's a good question, but you do have the genomic coordinates. So chromosome 1 to 22, you segment each chromosome one by one. So all the
or even correspond, because the columns correspond to the people and the rows correspond to
yeah.
So like, how are the people grouped together? Like, so that's that you have visible block column structure.
Oh, no, these are random. You can do some hierarchical clusterings of the individuals, and then they will be even clearer, I can promise you, but these are just random. And you can still see these, column-wise factors that pop up. Yeah, we don't group the individuals here. Yeah, and then we don't.
So that's feeling simply random community. Otherwise,
yeah.
You won't see a block.
Yeah, you don't see a block here. If I do a hierarchical clustering of either the rows or columns, it will be much cleaner than this. Yeah, and then we don't model the interperson correlations. I think if you do 500,000 individuals, especially they are all European Caucasians, you probably need to do some kind of relatedness model of the inter-individual covariation or correlation. Okay. But we don't do that.
Markov model, is this the linear Gaussian ID Markov model or is the linear non-Gaussian ID Markov model? What kind of ID Markov model that means?
A good question. Are you talking about the input data for the Hinomarkov model?
Not really clear, but I just heard you talk about
Yeah.
modeling.
Well, that is not necessarily modeling. We just take each row here. We do a segmentation for each of the individuals. And then for the Hinomarkov model, we can specify the transmission probabilities. That can have some biological bias come into it. And then the data is then segmented using the Hinomarkov model. You have different states. The states correspond to the copy numbers.
Is the state dynamic is Gaussian?
I'm not sure I understand your question, but the input data is Gaussian-like.
Model data is there is a state evolution, right? That can be Gaussian or non-Gaussian.
Maybe we can talk more about this. I'm not aware of that notion. Yeah, but in this case, maybe it's simpler than that. You have 5 different states correspond to the 5 different copy numbers. You have a Gaussian-like input. You specify the transmission probabilities. Okay, and then you just do the Hinomarkov model segmentation from there and on. It tells you the posterior probability of assigning each of the axon into one of the state. And then because it is sticky, you do have the jump, the transition states, and then you can segment this region into a consecutive the same copy number states. Yeah, maybe we can talk more about that later. Okay. But anyway, these are the independent benchmark results that tells you that Codex seems to have high sensitivity and specificity compared to the others. Until we apply this to some cancer genomics data. So in this case, it's a melanoma data. And then in this case, the blue colors correspond to the deletions and the orange color correspond to the duplications. Okay, so we talked to the oncologist. And then obviously we're missing a lot of recurrent duplications and deletions. For example, this BRAF is an oncogene that needs to be that are expected to be duplicated in most of the cancer genomic samples. We don't see it, but we don't see it. And then similarly, the P10 is a protective gene for the cancer evolution, and then we don't see its deletion. So it turns out that the common CNV signals are attenuated by the latent factors. Okay, so basically the latent factors not only capture the true artifact, not only capture the artifacts, but also they capture the common CNV signals. And then shown here, basically just a cartoon. You have some normal samples, you have some tumor samples. When I estimate the betas and the Gs, these actually capture some of the signals that I really want to retain in the cancer samples. Okay. Any idea how I can do a trick to improve this? Yeah. Exactly. I only use the normal samples to estimate my beta and Gs. I don't use all the samples, okay? Given that I do have the luxury to sequence some of the normal samples. And then a simple trick done here, yeah. Yeah. Well, for blood cancer, certainly that's the case, but melanoma is actually a skin cancer. I think it's easier for you to get that out. Yeah. So it's more pure compared to the liquid biopsies, for example. Yeah. So the simple trick, we can recover these duplications and deletions, and then you do see this very nice, clean stripe towards the bottom. Any idea what these are? Like white. Don't see anything. Those. Yeah, or my normal controls, right? So I use these or cancer free. Yeah. So basically, I use these to extract the latent factors. In the simple validation compared to an independent cohort shows that we are able to recover the correlation coefficient compared to this orthogonal validation source. But this is the case where we're able to find some normal controls. Okay, so after this is published, we talked to our collaborators. We're saying, okay, if you want to sequence the cancer genomes, throw in a bunch of blood samples as normal controls. So these can help you capture the noise terms. But in other cases, you just don't have the liberty or the luxury to include the normal controls in that case. What do you do? And you still see these common series that are, for example, presented in a cohort of individuals that are also very common. So what do you do? I'm not going to show you the details, but in this case, we're able to derive a EM algorithm, and then the hidden data in the EM algorithm basically is your carrier status of the copy numbers, okay? So instead of just fix the carrier status to be 0 and 1, I iteratively through the EM algorithm trying to give an estimate of that carrier status head, okay? And then without going into the details, these are the final figures for this paper. SVD, as you can see, the Z-scores cannot separate the deletion status from the null status. Now, if you use the negative control samples, so basically you take part of the samples, you're able to recover the true signals. Then we can also resort to the negative control regions, which are for the second case that I mentioned in the previous slide. You're also able to recover the true signals from the null status, okay? And then the validation certainly shows the case that if you look at the common CNVs, this enhanced version significantly boosts the performance in terms of both, recall and precision. So recall is just sensitivity. Precision is not necessarily specificity, but it's a fraction of the true positives outside of all the positives that you caught, okay? And then if you look at the rare ones, it seems that they don't differentiate that much because the rare ones tend not to be captured by shared factors across all the samples or across all the axons, okay? And then if you look at the common ones, it seems that is an average, performance compared to the common CNVs and the rare CNVs. But they also resort to some additional benchmark studies. Now, these are very important. I know that maybe for stat students, these seem tedious, but these are very important for you to publish in a very top, more high-profile genomics journals these days. And then the bar has just been higher and higher, okay? And then maybe just 4 samples, 4 methods you benchmark here is not sufficient. You know, an essay reviewer will probably say, okay, benchmark the other 2 for us. Again, I think sometimes it serves a purpose. And then you do see a lot of these benchmark studies actually published in Nature Methods and Nature Biotechnology these days. I think, especially if they're independent, okay, you don't have any bias. I think, given the fact that this is such a competitive field, you do have to have these independent opinion in terms of which methods I should use, especially from the point of view of a clinician or a biologist. Okay, I just want to, mention that this is probably an inevitable phase of the project if you want to work in genomics. Okay, so that is bulk DNA sequencing, and I'm going to move on to single cell DNA sequencing from here and on. There are 2 different technologies. The first one is more conventional in the sense you do have to manually isolate each individual cell. It's very laborious. Each cell will be its own reaction chamber. You do the amplification, you do the sequencing, so on and so forth. Maybe 2 or 3 years ago, 10x Genomics released this commercial kit that is almost like a black box that you just push your cells into one end of the black box. The other end of the box is a library prepped single cells with droplets, and the droplets capture the cells, and then it does all the reactions within the droplets. It seems to work really well until that they ran into some IP issues. They have to turn it off. And then, there's a non-commercialized version that I was told is essentially the same. It's called DLP plus, okay? So this is another thing that I find quite interesting in genomic research is that sometimes you have to, it's almost like a hit or miss. When we developed the methods for this section, we saw that 10x Genomics really is going to gain its popularity. And certainly it does. It did for a short period of time. And then they had this issue, and then it's not readily available to anybody else. So it turns out that the second part of the talk didn't gain as much attraction as we had hoped, just because I guess we betted it wrong. If we worked into in spatial transcriptomics, maybe we're not as late to the game as we are at this moment. Okay, so this is one thing that I want to mention, that for genomics research, you always have to look ahead, stay ahead of the curve if you want to really make some big impact. You want to develop something that is really useful to the community. Okay, but regardless, let's go through this. So this is the evolutionary trajectory of the copy number variation. Yeah. Yeah.
Single cell get 1 cell and you're sort of precisely DNA sequencing. How do you map? So then the bulk DNA is sort of like, if I have a lot of single cells, how do I map between the 2?
Between the single cell and the DNA
Yeah.
and the bulk?
Yes. How do they map between them?
Aggregate the single cells, that's your bulk.
Okay, so,
Yeah.
so then you have the, so can I, so that means you're always stating that you have two cells, and I DNA sequence that one, I will have different tumor both. The population of cells, more like a histogram,
Yeah.
Sort of a sample within the
Yeah. The bulk data is an average. The single cell is a distribution of the average. The mean of the distribution is what you observe by your bulk. Yeah.
So then, what's the cells? So both really mixed distribution of
Yeah.
single cells.
But that's a very good question. It does make sense for germline CNVs because all the cells are the same. You don't necessarily need to sequence your blood by each individual cell if all that I care is that whether I inherit some of the population-based copy number variations, or from an evolutionary biologist, all that I care is on the population level, what are the germline copy number changes. But if we're talking about cancer, that's a completely different story because each individual cell is different. Now, in this case, the single cell resolution does help you a lot. So shown here is just an evolutionary trajectory of the cancer. Starts with the normal cells and they get expanded, and you do acquire different types of mutations along the expansions, and you treat the cells, but it doesn't, kill all of them, and some of them got expanded and lead to metastases. Now, if you just do traditionally a bulk sequencing, it's an average, it's a mean. But if you do single cell sequencing, you do see this, evolutionary changes. Okay. And our goal here is just to use single cell DNA sequencing in the cancer genomic setting to profile the copy number changes. Again, this is in cancer because in germline, you don't need single cell. You don't need to waste the money or the efforts. You just pull all the cells because they are all the same, okay? And then the data that we started with is this breast cancer data. We have T10 and T16. Without going into the details of the biology, we know that T10 is more heterogeneous. It's what we call the polygenomic. T16, it is monogenomic in the sense that there's only one dominant clone that expands from the primary tumor to the metastases. Okay, so don't worry about the details. This one is different subpopulations of cancer. This one is just one unique subpopulation of cancer. We first use Gini index to identify the normal cells because inevitably you would pick up certain proportions of the normal cells, and we want to take advantage of that. In this case, we use a Gini index, which traditionally is an economic term, but here we can think of it as a robust measure of your standard deviation or the uniformity distribution of the reads along the genome. So, cancer cells have higher Gini index compared to the normal cells. So you can use this to identify some of the normal cells. Okay, and then once you identify the normal cells, what I ask my student to do first is just to apply Codex 2, okay? Use these normal cells as your controls. And then it does tell us these different subpopulations of the cancer samples. And then it seems that the T10 is indeed, a polygenomic tumor with 3 different distinct populations of cancer. And then the T16 is more homogeneously distributed across all the cells. I mean, all the cancer cells. But if you look at the normalized Z-scores in this case, it seems that they are both attenuated towards the null regions. They are not expected, the peaks or the moles are not where we are expecting them to be at. And the reason is because these cancer samples underwent a very significant copy number changes events, okay? So under a true case study, if you have a null normal cells, you fit a, Loess function or a smooth spline, and they can capture the GC content bias. But if you have significant disruptions of the genomes, like in this case, triple negative breast cancer, you have these deletions, you have the null case, you have the duplications. Now, in this case, you have multiple different states of the copy numbers. And if you just naively treat all the cells as a null case and you fit the GC content bias, obviously it's going to be a biased fit, okay? It captures, in this case, it's kind of in stock between, the 2 different states. Okay, sometimes it also has a very unstable tails in this case. And then this indeed is a question, for a lot of the, available methods. For example, this Nature Methods paper, you see they even have this in their figure 1, when you fit the GC content bias, obviously you have, not all the beings are actually the true null case. You should not do that. So what we end up doing is just to introduce this alpha IJ, and then it tells us, the copy number states, and then again, it's an EM algorithm. And because the cells are all homogeneous, and then all the copy numbers are integer valued. So in this case, you do have the ability to discretize the alpha ij in this case. If you have a bulk sample, then all the cells are mixed together. There's no way for you to distinguish those. Okay, and then you can write out the full probability distribution shown here, and then you just iterate it, the estimation procedure to get estimation of the alpha ij, and the alpha ij also directly tells you the carrier status of the copy number states. Okay, and then once you do that, it seems that we're able to identify the true states, and then for each of the state, we're able to fit correct FGC content bias curve in this case. Okay.
Just identify which points go with which grouping.
Yeah, and then that's your hidden data in your EM algorithm. Okay, and then once you do this, you are able to recover the z-score that is not distributed with the modes at the expected locations in this case. And then I think I'm going to skip this part, but this is just a cross-sample segmentation. Instead of segmenting each sample individually, all the cells are from the same individual, and then they share the same breakpoints. So you should probably come up with a method to jointly segment the genome instead of if we sequence 1,000 individuals, they all have different breakpoints, and then you can just segment them individually. Okay, and then these are the final output. And then the paper was published, and then these are the bioinformatic pipeline. And then these are the visualizations. Again, this is a polygenomic tumor. This is a monogenomic tumor. You find these different subpopulations in the tumor that is polygenomic. And then we also, again, like I said before, adopted some orthogonal validations to show that the estimations actually are more accurate if you account for the different states of the copy numbers. And then we also apply it to a mixture of data where you spiking around 10% of cancer cells and then 1% of the cancer cells into background of normal cells. You're able to identify the cancer cells correctly from the background of the normal cells in this case. So these are what I have for the copy number parts, and I do want to spend the next 10 minutes talking about the more exciting and ongoing lines of research that are in my lab. Any questions? Yeah.
How expensive these single cell studies? How many patients, separate people have like that type of data?
Well, 10x genomics is very readily available and accessible, but it's not anymore. So that's, again, it's just such a pity, but I do know that several labs are able to do this. Nick Neven's lab from MD Anderson, they do this on a daily basis. Yeah.
Use that finding early stage cancer.
Yeah, I think potentially you can, especially now people are talking about early cancer detection. You just draw a blood of cancer, draw a tube of blood. And then because the cancer cells shred all the DNAs and it floats around your blood, you're able to trace back some of the copy number states. That's what people use. Yeah.
Because